Good afternoon, folks. This is going to be our online content for this week. We are picking up where we left off in class, discussing the properties of muscular tissue in general and skeletal muscle in particular, taking a look at how skeletal muscle does what it does. And we're going to be focusing today on the structure of skeletal muscle that enables it to carry out the unique properties of contraction that allow you to move muscle around. Now, we left off in class the other day taking a look at the structure of skeletal muscle and some of its unique physical features that are going to make the ability of the muscle to contract possible. And I'm going to pull up a diagram here that I'm going to be referencing to show you a little bit about how that works. So bear with me 1 second while I screen share with you. And the diagram I'm about to show you is directly out of your handout for this week. So if you're following along with my handout on muscular tissue and its properties, then you can find this exact diagram in there for your reference. Okay, here we go. All right, so here we can see a cutaway diagram of a skeletal muscle fiber, a myofiber, which is another way of saying a skeletal muscle cell. As we said the other day, every myofiber or skeletal muscle cell is actually a long strand of individual muscle stem cells called myoblasts that fused together during embryonic development, giving you the long muscle fibers that we see in adult muscle. That's why the muscle is multinucleated, because the nuclei of the individual myoblasts are retained in the adult muscle fiber. Now, we can see in here several of the features that I was talking to you about in class the other day that your muscle fiber retains. Here you can see the sarcolemma, the external membrane on the outside of the muscle fiber. Here you can see the T-tubules extending down into the interior of the muscle fiber. At the places where they meet down on the inside, where my arrow is on the screen right now, you can see the terminal cisterna, which is going to be the place where we store the reservoirs of calcium that are going to be needed for the muscle to contract for reasons we'll get into as we go along today. Other things you can see going on here inside the muscle cell. There is cytoplasm in here. You're not seeing a lot of it because it's kind of squished between other structures. We call the cytoplasm of a muscle fiber the sarcoplasm. You can see nuclei, several of them, scattered throughout. Here's a nucleus, here's another nucleus, here's another nucleus. Rather than being in the center, they're squished up against the inside of the sarcolemma. We can see mitochondria, numerous mitochondria squeezed in between other structures. Lots of mitochondria needed, since, of course, muscle needs to generate a lot of ATP to power movement. And we're going to see the web-like internal membrane that we call the sarcoplasmic reticulum, or SR, which is the same thing as the endoplasmic reticulum that we find in any other kind of cell. And you can see that it's this kind of webby material that we see all throughout the interior of the cell. Now you can see that your sarcoplasmic reticulum is wrapped around these long, thick rods of protein. These long rods of motion proteins are referred to as myofibrils, and the myofibrils are going to be really the active agents of how the muscle moves. Myofibrils are assemblages of motion proteins, bundles of motion proteins that fill up most of the space inside the muscle fiber. If you look at them head on in cross-sectional view like this, you can see lots and lots of individual motion protein fibers kind of bundled together. You can see the sarcoplasmic reticulum wrapped around the outside of the whole myofibril. Now here we can see we're taking a myofibril, cutting it in half so we can see what's going on in its interior. We can see in its interior, it's quite neatly organized. There's a very tight arrangement of motion protein fibers. And the individual motion protein fibers that we see in a myofibril are called the myofilaments. And they come in a couple different types. We have thick filaments, which you can see as the darker lines in here, alternating with thin filaments, which are the paler lines that you see in here. And it's that alternating pattern of thick and thin filaments that results in the striations, the dark and light banding that you will see if you look at skeletal muscle under the microscope. Now, within the myofibril, you will see this neatly arranged alternating pattern of thick and thin filaments of motion proteins. And the thick and thin filaments are made out of different kinds of protein. And it does matter. We're going to talk about this a little bit here. The thick filaments are made out of a type of motion protein called myosin. Anytime you type this prefix myo, that is something to do with muscle. So myosin, like you see here. The thin filaments, the paler lines you see in here, are made out of three motion proteins: actin, troponin, and tropomyosin. They're arranged in these alternating bands, and they're anchored to these zigzaggy lines you see here called Z-discs. The space between two Z-discs, one unit of motion proteins found between two Z-discs, is called a sarcomere. Prefix sarco means body or mass. You'll see that term used a lot having to do with muscle, as you have seen. Sarcoplasmic reticulum, sarcolemma, sarcoplasm. And the suffix mere means unit. So it's kind of like unit of mass, unit of muscle. And the sarcomere, that unit of alternating thick and thin filaments of motion protein between the Z-discs, is really the contractile unit of the muscle. Each sarcomere is going to contract independently of each other sarcomere, and that is where the motion in the muscle is actually going to happen. And if you look here in our myofibril, here's a sarcomere between the two Z-discs, and you can see behind it, here's another sarcomere. And if you followed back along the whole length of these long myofibrils, there'd be one sarcomere after another, going all the way back along this rod of motion proteins. It's just one sarcomere unit following another. The thick filaments are anchored to the Z-discs, by the way, with another type of protein, elastic filaments, made of a protein called connectin or sometimes titin. You can use either name for that protein. So you find thick filaments, thin filaments, and elastic filaments making up your sarcomere. Now, the proteins in here, myosin and actin, those are the main ones. Those are what we call the contractile proteins. They're the main substance of those dark and light lines you see in there. Myosin and actin are the contractile proteins. They're the ones that actually do the action of the muscle contracting. When a muscle contracts, myosin and actin are the agents that make it do that. Troponin and tropomyosin, which are part of the thin filaments, are the regulatory proteins. They control when the muscle contracts. And finally, connectin, the elastic filaments, is what allows the muscle to spring back to its resting length after it's done contracting. There's a lot going on there, obviously. Now, to show you how all this works, obviously you can see kind of in miniature right here. Here are your Z-discs. Here are all those filaments arranged between. But let's zoom in on a sarcomere. Let's look at it a little bit more closely so you can see a little bit up close how this is all arranged. Here is a sarcomere kind of up close. At either end, here are the Z discs anchoring the whole thing together. And remember, myofibril is a line of sarcomeres, one after another after another. You can see anchored directly to the Z discs are the thin filaments. In this diagram, the thin filaments are the blue lines. And they stretch from the Z discs towards the center of the sarcomere. Now, the center of the sarcomere, see that line right down the middle here that I'm kind of highlighting at the moment, that's called the M line. That is the central point of the sarcomere. The thick filaments, which are the red ones in this diagram, originate from the M line. Pardon me, folks, a little congested here today. They reach from the M line towards either end, towards the Z discs. So thick filaments go from the M line in the middle towards the Z discs. You see the thick filaments, which are made of myosin, don't reach all the way to the Z disc. There's a little bit of space in here. That's not part of the thick filament you see right there. The thick filament is just all this red stuff, you know. That little bit you see right there that I'm kind of highlighting here at the moment, that's not part of the thick filaments. That's a little band of elastic filaments that anchors it in place. So the thick filaments start from the M line in the middle, but they don't go all the way to the Z disc at the end. The thin filaments, on the other hand, they start from the Z disc. See how that blue line originates from the Z disc, stretches towards the middle, but it doesn't reach all the way to the M line. So the thick and thin filaments overlap each other, but they don't quite make it all the way to the opposite ends of the sarcomere. Now you see a lot in my diagram here of terminology with lots of letters on here and everything. The space between the Z disc and the end of the thick filaments is called the I band. And that's the place where you have thin filaments, but not thick filaments. The thick filament doesn't quite reach that far. The place between the end of the thin filaments and the M line is called the H band. That's the place where you have thick filaments, but not thin filaments, because the thin filaments don't reach that far. The zone of overlap, thick filament overlapping thin filaments, that's the A band. And I know lots of letter terminology here. I didn't name all this. I'm sorry if it's confusing. So, what is going on here? What is going to actually happen? Why does all this matter? The reason all this matters, folks, is because of what's going to happen when the muscle contracts. Now, you see two different diagrams here. The one I've been describing to you is the top diagram here, and that is the muscle in its relaxed state. That's what a sarcomere looks like when it is relaxing, when the muscle is not yet contracted. When a muscle is contracting, here's what's going to happen. The sarcomere is going to change, and it's going to shift so it looks like the bottom diagram. Look what's happened in the bottom diagram. What's happened is that the thick and thin filaments slide over each other, and they overlap more than they did before. Basically, the whole thing squeezes inward. The H band and the I bands disappear because the thin filaments are pulled in towards the center. The thin filaments pull in towards the M line. So the ends of the thin filaments completely overlap the thick filaments. When that happens, the ends of the thick filaments reach the Z discs. And now you can see here in this bottom diagram, there is no more H band. The H band has closed up because now the thin filaments and the thick filaments completely overlap, and they both reach the M line. The H band has been obliterated. Likewise, when this happens, the thick filaments go all the way to the Z discs now. There is no more I band because the Z discs have been pulled inward, and the ends of the sarcomere have contracted inward. The entire sarcomere has gotten shorter, and its ends have pulled in towards the center. The I bands have disappeared as well, because now the thick filaments and the thin filaments completely over. So basically, both of those zones that we had where it was only thick filaments in the H band and only thin filaments in the I band have disappeared as the filaments have slid over each other. And now we have a shorter sarcomere in which the thick and thin filaments completely overlap. But when the whole thing shortens, the ends scunch inward. Scunch is a technical term. And that means that the Z discs pull closer, the whole sarcomere gets shorter. Now, that doesn't shorten the sarcomere itself by very much. But when you have a whole bunch of sarcomeres along the length of the myofibril, and each one after another, gets a little bit shorter, the entire myofibril gets shorter by a lot. So individual sarcomeres only shorten by a wee bit, but when you have a whole bunch of them, each shortening by a little bit, the entire muscle fiber can get shorter by as much as 40%. That's what's happening when a muscle contracts, folks. Muscle contraction is basically just thick filaments sliding over thin filaments, making the sarcomeres get shorter, one sarcomere after another, shortening the muscle until it pulls on whatever it is attached to. That's muscle contraction. And that's why I'm going into so much detail with this tiny little microscopic structure, because it's at that level of structure that we're actually seeing the action of the muscle be performed. So it's all well and good to say that's what is happening. That's the physical action that's taking place. That is our movement of thick filament overlapping thin filaments to make a muscle act. But what's actually mean? What's actually taking place? How does it occur? That's the thing we need to talk about next. Now, to talk about that, we're going to have to go into something else. We're going to have to start exploring how, in fact, the muscle gets stimulated in the first place. And so here we have to talk about the communication between your nervous system and your muscular system. How does the body tell the muscle to go? Now, we'll talk about what's called innervation of any given muscle. When we talk about the term innervation in anatomy refers to the nerve supply of any structure. And some structures receive more nerves than others. Muscles have a pretty high nerve supply for the most parts, but not all muscles receive equal numbers of nerves either. In general, the nerves that feed muscles, the nerves that go to your skeletal muscles and tell them what to do, are called somatic motor neurons. Now, as a somatic motor neuron approaches a muscle, It's going to branch extensively. And each branch of that neuron is going to touch one individual muscle fiber. It has to go to an individual muscle fiber because remember how every individual muscle fiber is wrapped up in a layer of connective tissue called an endomysium? Those endomysium layers of connective tissue act as insulators. They're kind of like the insulation wrapped around an electrical wire. They prevent the electrical signal from the nerve from going somewhere else. So if you want to stimulate that muscle fiber, it has to have its own individual little nerve fiber that goes to it. Now, that said, if you've got one nerve coming in and it branches and it puts out lots of little individual branches that go to several different muscle fibers, that means a signal coming down one nerve fiber can be distributed to multiple muscle fibers at once if all those branches go to a bunch of different individual muscle fibers. The term for this, for one nerve fiber and all the individual muscle fibers that it innervates, is we call that one motor units. Now, not all motor units are equally big or small. Some of them are different sizes. We can talk about small motor units versus large motor units. A small motor unit typically has fewer than 10 muscle fibers controlled by one nerve fiber. 3 to 6 is common. Now, if you have that few muscle fibers being controlled by a single nerve fiber, needless to say, that's not a lot of force. You can't generate a lot of strength with a small motor unit. It's very weak muscular contraction. But the trade-off for that is you get very fine-tuned, highly precise control. Small motor units are really good when the body wants to engage in very precise movements, very fine-tuned control, very small muscular action where we're moving the muscle just a wee little bit at a time, and we want to have very good precision movements, tiny little movements of things like eyelids or fingertips, things like that. So that's what small motor units are. Large motor units, you have a lot more muscle fibers per nerve fiber. Large motor units can get quite big. You might have as many as 1,000 muscle fibers per nerve fiber. Now, when you have that many muscle fibers per nerve fiber, you're generating a lot more power, a lot more strength in the contraction. You can engage more of the muscle at once. You're generating a more forceful muscular contraction. The trade-off for that is there's less precision. More power, more force, but less control. So you're going to find more of those large motor units in like muscles of your calves, muscles of your back, places where the precision isn't as much needed, but the force is required. Now, here's the thing that you need to know. Any given muscle in your body, a muscle is an organ, and any given muscle in your body is going to contain multiple motor units. You don't contract the entire muscle at once. There are going to be several motor units in each muscle. And the way that this will work is that when you are engaging that muscle, your nervous system will send signals to that muscle and will activate motor units and generate some force. And if that force is adequate to move the load, that's all we'll do. Our bodies try to conserve energy, folks, and they don't spend more than they have to. As I've said in here before, I think movement is expensive. Movement is the most expensive thing a body does. It takes a lot of energy, a lot of ATP. And so to conserve energy, our bodies try not to burn more force than they have to. They try not to spend more ATP per movement than is necessary. So when your body activates a muscle, Basically, what it's going to do is it's going to activate motor units one by one until it activates as many as are needed to shift the force that shift the load that it's trying to shift. You don't feel this happening very much, but when you're trying to shift a load, the muscle will get more and more and more tension in it. And that's because more and more and more motor units are being engaged until you have built up enough strength, enough force in the muscle to overcome the resistance of whatever load you're trying to move. At that point, we'll stop activating more motor units. We'll only use as many motor units in the muscle as are required to do the job being done. That's called recruitments, the process by which our muscles engage motor units one by one by one until we have enough power to do the job that needs to be done. That does two things. One, it allows us to conserve energy by not engaging more than we have to. And the other thing that lets us do is it means that if we're doing sustained work, if we're bearing the load for a while, and we're not engaging the entire muscle at once, we can actually shift off between motor units. That if it's a sustained activity, if there are some motor units that are not yet being engaged, we can actually let individual motor units rest. That while you're engaged in sustained labor, individual motor units will switch off. Some of them will relax, and other ones will take up the slack that haven't been activated yet. And that allows different parts of the muscle to alternately relax, and then other parts take over. That's a defense of the muscle against fatigue. And that allows the muscle to go for longer by having different motor units within the same muscle take turns bearing part of the work. Now, in order to make this happen in the first place, we have to get a signal to the muscle from the nerve. And that means we have to have communication between the nervous system and the muscular system. The interface between a nerve and a muscle is called the neuromuscular junction. And we can see a neuromuscular junction illustrated right here. Now, a little bit about what you're seeing here. This side of the neuromuscular junction, this is the neuron. This is the end of the nerve fiber where the signal is coming down. This side of the neuromuscular junction is the muscle fiber that's being stimulated by that nerve. Now, to show you what's going on here, you see that wavy line here? That is the surface of the muscle fiber, which at a microscopic level is not a nice smooth straight line. It's kind of jaggedy. That's our sarcolemma, the membrane of the muscle fiber. You can see that all throughout its surface, those little kind of tube-looking things, those are ACH receptors. Those are receptors for signals from the muscle, from the nerve. This is the end of our nerve fiber. At the end of the nerve fiber, and the nerve fiber is called an axon. We're going to talk more about that in the next chapter when we talk about the nervous system. And at the end of the nerve fiber, the axon swells into this kind of button-shaped arrangement called the axon terminal. And the axon terminal is going to be the end point of the nerve fiber. The interface between the axon terminal and whatever structure it's sending a signal to, whether it's a muscle, whether it's an organ, whatever a nerve is stimulating, is called a synapse. That's a term you're going to hear a lot in neurology, but the synapse is the interface between a nerve fiber and whatever it's stimulating. And the word synapse is both a noun and a verb. You can say the synapse is the point of contact. You can also say this nerve synapses with this muscle or with this gland or whatever. Now check it out. The synapse is actually not a point of contact. There is a little tiny gap between the nerve fiber and the muscle fiber. It's not a very wide gap. It's about 60 to 100 nanometers in diameter. We call it the synaptic cleft. A cleft is a gap if you don't know that word. We refer to the nerve on this side as the presynaptic neuron, presynaptic before the synapse, or the presynaptic cell, or the presynaptic nerve fiber. You can use any of those terms. We refer to the muscle on the other side as the postsynaptic cell or the postsynaptic fiber, whatever term you want to use. Now, you might be thinking to yourself, okay, electrical currents can't travel across empty space. They require a continuous circuit. They require continuous contact to move. How do they get across the gap? Here's how it works. So our electrical signal from the nerve comes zapping down the gap, arrives at the axon terminal. Here in the axon terminal, there are going to be vesicles containing molecules known as neurotransmitters. Probably all heard that term before. Neurotransmitters are the chemicals that the nervous system uses to communicate. And there are a lot of different kinds of neurotransmitters. Some of them are used exclusively within the brain. Some of them are used in the body as a whole. And we're going to talk more about neurotransmitters when we talk about the nervous system in the next chapter. Different kinds of neurotransmitters are used for sending different kinds of signals. The neurotransmitter we care about right now is a neurotransmitter called acetylcholine. Abbreviated ACH. Capital A, capital C, little h is the standard abbreviation used in neurology for that. ACH is the specific neurotransmitter of muscle. It is used by neurons throughout the body to communicate with muscles. So, in our muscular system, the axon terminals have vesicles filled up with ACH, with acetylcholine. That's the neurotransmitter that stimulates the muscle to action. When that electrical signal arrives at the axon terminal, that will stimulate exocytosis, causes these vesicles to fuse with the membrane of the axon terminal and release ACH into the synaptic clefts. These little bead-like things you see in the diagram here are molecules of ACH. The ACH molecules are released into the synaptic clefts, travel across that gap, and binds to the ACH receptors located on the postsynaptic muscle fiber. When they do that, that initiates the chemical process on the far side that stimulates the muscle to action. As long as the ACH receptors are receiving signals from ACH, the muscle will be stimulated to act. When the nerve is no longer stimulating that action, exocytosis of ACH stops. At that time, an enzyme in this gap called acetylcholine esterase, bit of a mouthful there, we typically abbreviate it ACHE, capital A, capital C, little h, capital E. That enzyme will enter the synaptic cleft and clean up. It will break down ACH in the synaptic cleft. Once the ACH is no longer present, the muscle is no longer being stimulated, and muscular action will stop. This is all very rapid. So when I move a muscle, this all happens really quite quickly. ACH is released, stimulates the muscular action, muscle acts. I stop moving the muscle, ACH stops being released, ACHE moves in, breaks down the ACH in the synaptic cleft. The muscle is no longer being stimulated, muscle relaxes. That whole thing takes place within a fraction of a second. There are things that can go wrong here. In an individual with inadequate ACH receptors, you have a condition called myasthenia gravis, in which it's difficult for the nerves to stimulate the muscle. And that can happen if there are inadequate ACH receptors, or if those receptors are defective and not binding ACH properly. Certain neurotoxins can bind to the ACH receptor and cover it up, preventing ACH from reaching it. There's a terrible nerve gas called sarin. Was used as a weapon of war at one time, that blocks ACH receptors, leads to paralysis in the victim, and actually leads them to suffocate because their lungs cannot be ventilated, because they can't move the muscles in the chest that ventilate the lungs, and they basically suffocate to death because they can't breathe. There was a terrible nerve gas attack on a subway in Tokyo in the early 2000s when a terrorist group released sarin gas in a crowded subway car during a Tokyo rush hour, and killed a couple dozen people back in the early 2000s. If the ACHE enzyme fails, you may find that ACH cannot be cleaned up. Muscles will tighten and contract, but will be unable to relax. That's what happens in somebody who's suffering from tetanus. That's or what we call that spastic paralysis. That's the formal name of that. Basically, failure of ACHE. And somebody who's got tetanus, that's exactly what's happening. That the ACHE enzyme is failing, and therefore muscles will tighten and be unable to relax. And here's a little fun fact for you. A couple of drugs that we commonly use bind to the same receptor sites as ACH and can therefore stimulate muscular action even in the absence of other stimulus, namely caffeine and nicotine, which is why those drugs can give you the jitters. Okay, so that's how a nerve sends a signal to a muscle. Making sense? I'm asking this if you're here. Okay. So having said that, we have now laid out kind of the pathway here. We've seen what the parts of the muscle are that are going to be actually engaging in contraction. We've seen how the nerve is set up to communicate with the muscle. Now we know everything we need to know to talk about the process of muscular contraction. So let's go through this step by step and see what actually takes place during a muscular contraction. How does a muscle, in fact, contract? Now, the process of muscular contraction is a four-step process. There are four distinct stages that a muscle goes through when it contracts. So let's go through them one by one. And I know this is a highly technical process, so just bear with me here, follow along. It's all written down in your note packet for today, and I'll be more than happy to review this with you in class next time I see you if you're not sure about any of these details. But let's begin. First part of this, excitation. Excitation is the phase at which we are going to send an electrical signal to the muscle to stimulate it to action. During the excitation phase, a nerve impulse arrives at the axon terminal and sends a signal across the gap exactly in the way I have just described. ACH travels across the synaptic cleft and binds to ACH receptors on the postsynaptic muscle fiber. So what happens once that occurs? First off, you have to understand a thing. Your muscles always have a low-grade electrical charge. I think I talked about this a little bit in class the other day, but let's remind you about this detail, that a muscle fiber at rest has a low-grade electrical charge that we call its resting membrane potential, or RMP for short. And I may have given you the wrong number about this the other day. I can't recall. Did I say negative 60 or negative 90 millivolts? I don't remember in class. But a muscle fiber at rest has a resting membrane potential, or RMP, of approximately negative 90 millivolts. In other words, low grade negative electrical charge. It's a static charge. It's not changing. It's just kind of sitting there. So your muscles always have a slight negative electrical charge when you're not doing anything with them. Now, here's what happens. When ACH binds to the muscle fiber, that stimulates the opening of gated ion channels on the surface of the muscle fiber. Specifically, it opens up channels that admit extracellular sodium into the muscle. Now, sodium as an ion is positively charged. So when extracellular sodium flows into the muscle fiber, that causes the charge of the muscle to go from negative to positive quite rapidly. And the muscle's charge rises from negative 90 millivolts to positive 75 millivolts very quickly. Now, as soon as the charge hits positive 75 millivolts, that threshold charge triggers the opening of another set of ion channels. These are ion channels for potassium. And when those channels are open, potassium ions inside the muscle fiber rush out. Sodium rushed in a moment ago, potassium is going to rush out. Now, potassium is also positively charged. So when a whole bunch of potassium rushes out of the muscle fiber, the electrical charge of the fiber is going to go down again. And it's going to drop back down to negative 90 millivolts, restoring the resting membrane potential to its original level. That rapid shift in the electrical charge from negative 90 to positive 75 back down to negative 90 is going to produce what we call an action potential. In other words, rather than just being a static, unchanging charge on the surface of the muscle membrane, it's going to produce a moving electrical charge, a wave of stimulus that went from negative to positive to negative. Now we have this wave of electrical stimulus that's going to travel along the length of the muscle fiber. That's excitation. We would say the muscle is at this point excited. So that's step one. We have put the muscle fiber into an electrically excited state. And by the way, if you are wondering, you might be thinking to yourself, okay, well, if we send all this sodium in and send all this potassium out, how do we get those things back to normal so that that can happen again? That's what your sodium-potassium pump is for. Okay, step two. Now that we have excited the muscle, we go into what's called excitation-contraction coupling. That electrical charge that we have just generated is going to travel down the length of the muscle fiber. Now, initially, it's just on the surface of the sarcolemma. It's on the external membrane. But remember our T-tubules that go down into the interior of the muscle? Those T-tubules are going to conduct that electrical charge down into the interior of the muscle fiber. The T-tubules are going to be connected to the terminal cisternae, which you might recall is where the calcium ions are stored. When that electrical charge is conducted down into the interior of the muscle fiber, that stimulates the release of ionic calcium from the terminal cisternae into the cytosol of the muscle fiber. Now that there's free calcium circulating inside the muscle fiber, calcium ions are going to bind to the troponin proteins found on the thin filaments of our myofibrils. Remember I said that the thin filaments of the myofibrils consist of actin, troponin, and tropomyosin. Troponin has a binding site for calcium. And now that there's free calcium circulating, calcium is going to bind to the site on troponin. When it binds calcium, that causes troponin and tropomyosin, which we call the troponin-tropomyosin complex, to shift and change position on the thin filaments. Under normal circumstances, our thin filaments are non-polar. They're just sitting there. They're not able to interact with the thick filaments of the sarcomere. They're non-polar. They don't interact with anything. They don't stick to anything. They have polarized sites on them, however. The actin fibers of the thin filaments have polarized spots on them called active sites. But those are normally covered by the troponin tropomyosin complex, preventing them from sticking to the thick filaments in the sarcomere. Remember how the thick and thin filaments overlap each other, but they're alternating with each other? Normally, they don't interact until now. When calcium binds to the troponin tropomyosin complex, that complex shifts out of the way, uncovering the active sites on the actin fiber. Now, that polarized spot on the thin filament is exposed, and it's able to potentially interact with the thick filaments. Now, we are ready to get this sarcomere into action. So in summary, what happened during stage 2 here, excitation, contraction, coupling. The action potential, the electrical charge that we generated during step 1, travels down the length of the muscle fiber, is conducted into the interior of the muscle fiber by a T-tubule. That stimulates the release of calcium from the terminal cisternae. Calcium ions then binds to the troponin in the thin filaments. This causes troponin and tropomyosin in the thin filaments to shift position, which exposes polarized active sites on the actin thin filaments. And now that those polarized active sites on the thin filaments are exposed, the thin filament has a way to interact with the myosin thick filaments. So that's our step 2, excitation, contraction, coupling. That brings us to step 3, contraction. Now that we've got all this going on here. We are ready to make this thing contract. Our thick filaments of myosin are going to bind and break down or hydrolyze a molecule of ATP. That provides the thick filament with energy. We say the thick filament is being, quote unquote, cocked into a high energy position, like you're cocking a gun. Now, if you remember when we looked at our diagram before the sarcomere, actually, I'm going to pull up something that I didn't point out to you before when we were looking at our diagram, but give me a moment here. Look at our diagram. Look at the thick filaments. Look at those big bulgy lumps on the thick filaments. Those are called the myosin heads. They are highly polar too, much like the active sites on the thin filaments. What's going to happen is those polarized myosin heads are going to bind to the active sites on the actin thin filaments. When they do, they're going to flex into a lower energy position. We call that the power stroke of the muscle. When they do that, they pull the thin filaments along with them. This causes the thick and thin filaments to slide over each other and pulls the Z discs closer in towards the center, just like we described before. So it all slides inward and makes the entire sarcomere shorter. Now, here's the thing. The filaments themselves don't actually shorten. The thin filaments just move over the thick filament. The length is retained. We just made them overlap each other a bit more. Once they have slid over each other in that way, they remain in that flexed position until they bind another molecule of ATP and release the actin fiber. More energy is actually required to let the muscle go and relax it. Relaxation takes energy. It's not just a matter of tension is not automatically released. The act of releasing the energy is called the recovery stroke of the muscle. That is, quote unquote, recocking the muscle to put it back to its resting position. And remember, this happens in every individual sarcomere along the length of the muscle fiber. Individual sarcomeres might only shorten the muscle fiber by 1% each or less, but their collective action might shorten the entire muscle fiber by as much as 40% total. So that's contraction, folks. And in summary, what happened here is myosin bound a molecule of ATP to get energy. Myosin and actin attached to each other. The active site on the actin bound to the myosin heads. Used that energy to slide over each other and overlap, pulling the ends of the sarcomere inward and causing the entire thing to get shorter. That's our power stroke, and it's going to stay that way until more ATP is bound to release it, which is called the recovery stroke. That whole thing, by the way, that action of movement that I just described is sometimes referred to in muscle physiology texts as quote unquote sliding filament theory to describe the action of how the muscle works. Now, the final thing that needs to happen here, of course, is relaxation. After all, muscle can't go again until we put it back to its resting position. In the relaxation phase, nerve impulses stop coming in. ACH stops being released by the neuron. The enzyme ACHE moves into the synaptic cleft, breaks down the ACH that's in there, and the muscle stops being stimulated. As a result, calcium inside the muscle fiber is pumped back into the terminal cisternae and stored by a protein called calsequestrin. When calcium separates from troponin, the troponin and tropomyosin shift back into position, recovering the active sites of the thin filaments. When that happens, myosin can no longer bind to actin. And when you do that, the tension is released. The fibers let go of each other, tension goes, and the elastic filaments that I mentioned before cause everything to spring back to its original position. As a result, the muscle is restored to its resting length, and the sarcomere goes back to its original position. Sometimes when this happens, another muscle kicks in. A lot of times when one muscle relaxes, another muscle will flex. We'll call that an antagonistic muscle. And you'll find a lot of situations in your body that one muscle relaxing will cause another muscle to activate. A lot of muscles have antagonists that work in pairs like that. Now, I should point out, there are times when we will have made a muscle go, and then we'll try to make it go again. You'll stretch, and then you'll try to stretch again from the same position you're already stretched from. If you're trying to stretch a muscle that's already partly stretched, you can do that, but the thick and thin filaments have less room to slide. As a result, we will say the muscle will have poor muscle tone. Optimal resting length is needed for the highest muscle tone and generating maximal force. Therefore, a muscle that is already partially stretched will have poor tone and will exhibit a weaker contraction. Therefore, it won't generate as much force, and you'll be more likely to injure yourself whenever you're trying to act with that muscle. A classic example of this, everybody always tells you when you're trying to pick up a heavy object, lift with your legs, not with your back, right? Reason for that? If you bend over to lift a heavy object from the waist, the muscles of your back are already stretched. So if you try to lift that heavy object, they're going from a less optimal position. They have less ability to exert force. Their tone is poor. You're more likely to pull them and damage them. On the other hand, if you are squatting down to pick up heavy objects, the muscles of your back are not already in a flexed position. They're at optimal resting length. They can exert maximum strength. And the antagonistic muscles in your legs are flexed into a relaxed position, which means that when you unflex them, they can exert maximum strength to stand you up again, and therefore you can get all that power to work with. Make sense? Now, because it's spooky season, let me introduce you to a little cool detail here. The process of pumping the calcium back into the terminal cisternae to relax the muscle is an active transport process. Like I said, it takes energy to relax the muscle. It's not free. You have to spend ATP for that. When you die, as the body begins to decay, the terminal cisternae begin to break down. As the terminal cisternae begin to break down when a body is decaying, calcium is released into the muscle fiber. The release of calcium into the muscle fiber of a decaying body causes those muscles to gain tension, even though you're dead, because the calcium still binds to troponin. It still shifts and exposes the active site on the actin. The actin still binds to the myosin, and the sarcomeres tighten up, which means that a dead body can gain tension in its muscles. We call that rigor mortis. And you all have heard of that before, that a dead body can gain stiffness and tension, and therefore a little bit after it begins to break down and decay, you'll find that a dead body will stiffen up. That's because calcium has been released from some of the terminal cisternae and is making the muscles do what they do. But because there's no ATP being produced by a dead body, there's no active transport process to reabsorb that calcium and nothing to make that tension release. So the dead body stiffens up and it doesn't relax. So something like 8 to 12 hours after death, a corpse becomes stiff and rigid. And that process of rigor mortis actually is useful in forensic science for helping to estimate, depending on the temperature of the environment where the corpse is, it sets in at a certain period of time, how long somebody's been dead. It also makes things a really real pain sometimes in forensic scenes, trying to move a dead body, because a dead body with rigor set in becomes very stiff and rigid and hard to move. I speak from experience, having worked in forensic science. Also because when it sets in, sometimes muscles will move. You might have all heard this before, and there's the spooky season part. Dead bodies move sometimes. Sometimes they will twitch when rigor mortis begins to set in and a muscle stiffens. Sometimes dead bodies will exhibit little twitches because muscles will go a little bit. They'll twitch, limbs will bend. If muscles in the chest cavity twitch, air will be pushed over vocal cords, and sometimes a dead body will moan. If muscles in the back twitch and go into rigor mortis while other parts of the body are still loose and not in rigor yet, because it doesn't happen to the whole corpse at once, and the body's at the right angle, sometimes the back muscles will tighten and the corpse will sit up. Sometimes rigor will happen in the hand and the fingers will curl. And let me tell you, when you're in your forensics cadaver lab and you put your hand down near the hand of the cadaver, right when the cadaver goes into rigor mortis and the corpse tries to grab your hand, that is a moment you never, ever, ever forget. Happy Halloween. Okay. So one last thing here. Whenever the body is doing all this, like I said, you don't gain all of the tension at once. Muscles do not build up all the force they need immediately. When you're starting to make a muscle go, there's going to be a very quick, rapid cycle of contraction and relaxation that we call a twitch. It's the minimum stimulus needed to generate an action potential, the threshold, as we call it. Individual twitches are too weak to do any significant amount of work. But what's going to happen is the muscle is going to say, all right, we need to build up tension. We need to get enough tension to overcome the force of whatever load it is we are trying to make go. And so when you're trying to make muscles work, the nerve will stimulate the muscle and a twitch will happen. And then more motor units will need to be activated. So another stimulus will come in. Another twitch will happen. and another motor unit will be activated, and another twitch will happen, and more will come in until sufficient tension is generated to make the muscle go. That is referred to as multiple motor unit summation, also known as MMU summation, or the name I gave for you earlier is recruitment. And therefore, any muscular action is an accumulation of twitches until the muscle has built up adequate tension to overcome the resistance of whatever load it is trying to do. You don't notice the individual twitches coming in because they're too quick. A typical twitch lasts for like 1/20th of a second. And they're just building and building and building until that comes in. Each twitch raises the overall tension in the muscle, which is called the trep of the muscle, TREPPE. The muscle never fully relaxes between twitches, and therefore more and more tension builds up, strengthening the tension in the muscle with each twitch until you have enough tension to overcome the force of the load. That phase is called isometric contraction, in which the tension of the muscle is less than the weight of the load. The muscle is gaining tension but not changing length. Once the tension overcomes the resistance, the muscle moves into what's called isotonic contraction, in which it changes length but does not gain subsequent tension because, like I said, the body conserves energy. It only activates as many motor units as are needed to overcome the resistance of the load so as to conserve energy and to allow motor units to switch off so that they can relax and avoid accumulating fatigue. A typical muscle in your body can safely handle 10 to 20 twitches per second. That's about normal. And that's usually enough to build up the tension you need to overcome the resistance of a normal load. Most muscles in your body under normal conditions, if things are going well, will not twitch at a rate of higher than that. Natural twitches don't typically come in at a rate of any greater than 25 per second. But once you have twitches coming in at a rate greater than 20 per second, 20 to 40 twitches per second, something begins to happen. If twitches are coming in at a rate of 20 to 40 per second, at that point, a new twitch arrives before the last one fully resolved. And that results in the muscle going into a state of what we call incomplete tetanus, in which the muscle begins to cramp up. The muscle can spasm a little bit and doesn't fully relax between twitches. It's like a painful spasm in the muscle, uncomfortable, starting to cramp a little bit. Can happen due to nutrient deficiencies, can happen when you're overworking the muscle or whenever it's twitching from an uncomfortable position. Under rare occasions, if the twitches come up at a rate of 40 to 50 per second, which is about the highest you'll get under nature, the muscle can go into a state of complete tetanus, in which all the twitches fuse into one big solid twitch. Muscle generates up to 4 times the tension it normally would, and it cramps up very painfully. When you get a very painful, solid muscle cramp, a Charlie horse or something like that, the muscle is in a state of complete tetanus. And at that point, the muscle locks up and is unable to do effective work because basically the twitches have come in too hard and too fast, and the muscle locks. And that's when you get a painful cramp in the muscle. Okay? All right, folks. So that's what I wanted to talk about today concerning muscular action. We are going to have more that we want to say about this. Next time we get together, we're going to want to start talking about muscle physiology. We're going to start learning about how muscles do what they do at a physiologic level. We're going to start learning about how muscles generate their energy, what happens when a muscle becomes fatigued, different kinds of muscle in terms of strength versus endurance, how we build muscle, how we lose muscle, and a little bit about the limits of muscular action. But for now, we learned about how muscles act at a basic level. And you now have the information needed for your first muscle quiz. So I'm going to post this lesson. And after I post this lesson, I'm going to be posting a quiz on the fundamentals of muscular action. Everything we talked about in class the other day is fair game for that quiz. Everything from this lesson is fair game for this quiz. So the basic properties of muscle, the organization of muscle, the cellular structure of muscle, the neuromuscular junction, the action of muscular contraction, the organization of the sarcomere, uh, everything I just discussed about how muscles gain tension, all of that is material that could be covered on this quiz. You'll have a week in which to complete it. Of course, you can take hold off if you want to hold off until after we talk on Wednesday, so you can ask me questions about it. That's completely understandable, but you will have until midnight next Sunday to finish it. All right. I will see you all in class on Wednesday. Take care.